Hello, everybody. Welcome to our webinar, Agricultural Fungicides and Public Health. My name is Dale Wehoff. I'm the Vice President for Communications at the Institute for Agriculture and Trade Policy, and I'll be hosting our webinar today. I just want to cover a couple of housekeeping items before we start the program. I want to let you know that we'll be recording the event, and following the webinar, we'll be sending out links to that recording and any other articles or references that we have will include that might come up during the webinar. And then also I want to encourage you to submit questions. We'll be tracking these questions as the webinar goes on, and once both of our presenters have completed their presentations, we'll start going through the questions. And if you're seeing my screen, which I hope you are, you'll see how you can ask a question. So the issue of fungicide and fungicide resistance is relatively new to IATP, and for me it started several years ago when I met a helicopter pilot who told me he was spraying fungicide on corn just as it was starting to tassel to increase yields. And I hadn't heard of this, and I started to be curious about how this worked and how much fungicide was being sprayed. And I was fortunate to shortly afterwards come across a paper from the organization of one of our presenters, the Center for Agriculture and Environment in Holland, called CLM. And they had prepared a paper called Resistant Fungi Are a Ticking Time Bomb. And that definitely got my attention. And so we began to collect stories and started a small monthly newsletter called Fungicide Watch. And this led us to preparing some reports, which we'll talk about as we go on. And we've continued to share information with CLM, and we'll learn from our presenter this morning what they've been doing. So I'd like to get into the presentations now, and we'll begin with Logan Luce. Logan is the author of IATP's new report, Fungicide Resistance, Risk and Consequences in Modern Agriculture. Logan is a Minneapolis-based ecological consultant. He has conducted ecological research in agricultural landscapes across the upper Midwest and advises IATP on the developing issues related to fungicide resistance. I'd like to turn the presentation over to Logan, and Logan will be followed by Peter Lendester from CLM, but I'll introduce Peter next.
Thank you, Dale. It's great to be here. Thank you all for joining us. If you want to understand fungicide resistance and the issues that are inherent and the public health issues that are related to this, first you really need to wrap your head around what fungi are. So that's where I'm going to be starting today. So fungi are one of the three main kingdoms of multicellular life here on this planet, along with animals and plants. And they have a really unique body plan. So instead of keeping all their cells together in one body, instead fungi are dispersed. They have many filamentous, what are called hyphae, which are combined together into a mycelial network. And these mycelia are ubiquitous. They're all around us in the soil and the water. And this body plan gives them a really great ability to decompose dead organic plants and animals. But, of course, it's also useful if you don't want to make your living on dead matter. You can also parasitize living organisms. So what you're seeing on your left is a mycelial network. And again, these are all around us. And on your right is a parasitic fungi. So this is coffee rust, which has been causing pretty serious problems in Central America in the past few years. But of course, we see fungi not as mycelia, but as mushrooms, right, and morels, and a whole variety of structures that are recognizable to us at a macroscopic scale. These are reproductive structures, and fungi are really excellent at producing really massive amounts of spores that are highly resilient to environmental stresses. So in addition to being ubiquitous in soil and water, their spores are also ubiquitous in the atmosphere around us. So of course you see a morel on the left, and on the right, that's a microscopic reproductive structure. Those are conidiospores of Aspergillus, which we will be hearing a lot about coming up here. The fungi have really diverse reproductive life cycles, and this is important because they can reproduce sexually or asexually, which gives them a really great diversity. And right, those reproductive structures are both most of our fungal food. They're what we eat when we eat fungi, but they also are really important for spreading pathogens, which can infect our food and indeed ourselves. So what do we do with those fungal pathogens? Well, recently we have been using fungicides, chemicals that selectively destroy fungi. And since the end of the 19th century, these have been a commercial commodity. So the first fungicide that was sold commercially was the Bordeaux mixture, copper sulfate, and you can see a ball and stick diagram of that on your lower left. This is, of course, highly toxic, and you need to apply it in large doses to really get any effect. We have come a long way since then, and in the 60s and 70s, much more modern, complex fungicides were introduced. On the upper right, you can see Benamil, which is a benzimidazole fungicide that was really the first fungicide that had resistance problems. It was introduced by DuPont, and really just a couple years after its introduction, they started getting fairly serious reports of resistance, and instead of dealing with those and putting out reports, they dealt with them on a very ad hoc basis. And because of the main serious problems with that, that response, fungicide companies came together in the middle 80s, I believe, and created the Fungicide Resistance Action Committee, or FRAC, which now does a lot of very serious work about fungicide resistance, but it is, of course, run by various fungicide companies. On the lower left, you see tebuconazole, which is a modern recent fungicide in the azole family, which again, we will be hearing a lot more about here shortly. So a recent review showed that more than 50%, more than half of agricultural diseases and the crops that they are found in are having resistance problems with their fungicides. So what is this resistance, and how do we understand both what it is and therefore how we can solve it and prevent it from happening in the first place? So if you have a population of fungal cells and spores in your crops, there's going to be genetic diversity inherent in that population. And because fungi are producing tens of millions of spores fairly regularly, because their mycelial networks are made up of millions upon millions of cells, there are going to be mutations, and some of those mutations are going to give an upper hand in terms of resisting the effects of a fungicide. Maybe they can exude the chemicals out of their cells so they never get to interact with them. Maybe the target of those fungicides changes so that the fungicide can no longer act upon them. There are a variety of different methods. Oftentimes, many of them are working together. But once a resistant individual appears, then whenever fungicide is applied, that will be a selection event. And so susceptible fungi, sensitive individuals, will get killed by that fungicide and allow those resistant individuals to spread further into the population. And each subsequent fungicide application is going to be another selection event, which means that we're going to have increasing problems with each fungicide application. So with this information, we can then judge which issues, which fungicides, which fungi, and which fungicide use application mechanisms are going to cause the most resistance problems. So if there's a fungicide that produces a particularly exceptional amount of spores, we know that that is a good indication that resistance problems are more likely. If you apply your fungicide very, very regularly, you know that that is going to increase the likelihood of resistance. And when you combine all those things together, you can create a pretty good picture of how fungicide is going to spread through a population. So this is real data about wheat powdery mildews and their resistance to a DMI fungicide that's in the same family as azoles, triademinol. And the height of these bars that you can see across Northern Europe are the resistance factors. Higher numbers mean that the fungicide that they've collected from those sites will require a much higher dosage of fungicide to see the same results. So you can see down in northern Spain, those populations are essentially completely similar to the wild type susceptible fungi. But up in northern France and the Netherlands, and you see much higher bars, and those are showing that because fungicides are often used in those wheat populations, they have a much bigger problem with resistance. So why do we care about this resistance? It obviously affects our crops, but why is this an important problem now? And in researching this with Dale and IATP last year, we found that one of the real issues is that there's a real incredible sort of explosion of emerging fungal diseases right at this point in history. And these are affecting animals and plants and indeed humans. So we've got a bunch of data on this screen. And in the left, you can see the growth of alerts on a major medical sort of newsletter, and the number of alerts for fungi over the past 20 years has really increased, both for fungi that infect plants and for fungi that infect animals and humans. And these fungi are not causing minor problems. Fungi have a lot of characteristics that make them particularly excellent at causing extinction or extirpation level events. So completely removing the host from the environment in which they are. And you can see a comparison in the pie chart in the center of fungi compared to other diseases. And you can see they contribute the vast majority of the extinctions in the past number of years. Moreover, you can see at the far right that those extinctions are increasing dramatically through time. And a variety of human drivers are causing that. Climate change is a big one. Land use change, turning natural ecosystems into cropland or pasture is a part of that. But one of the biggest ones, and one of the ones that I think IATP has hopefully some chance of helping with, is global trade, which can move pathogenous fungi into populations that are not used to them and introduce new and indeed fairly terrifying diseases. So a human example of that would be Cryptococcus gattai, which is a fungi native to Southeast Asia, which has recently in the past 20 years come to the west coast of North America and caused a large number of infections and indeed a couple dozen deaths. And this is not just related to humans and our crops. It's also in wild species. So a few little vignettes about these emergent fungal diseases before I turn all of this over to Peter, and he can give us a better example of how fungal diseases and fungicide resistance affect humans. So in the center of your screen right now, you see some bats that are infected with white nose syndrome, which is a very serious fungal disease imported from Eurasia since 2006, and it will likely cause the extinction of a variety of bat species, and that's going to cause major ecological problems for northern North America. On the left, you can see wheat stem rust, which is a worldwide issue. It's on 6 continents, and one of the strains of this is particularly good at destroying resistant wheat varieties, and it can cause 100% crop losses in some instances. On the right, there's a picture of a coral in a coral reef. This is a sea fan, and it is infected with Aspergillus, a very common genus of fungi that we will again be hearing about here very shortly. And these infections seem to be tied with problems that the coral is facing for other reasons. High temperatures because of climate change, changes in acidity, also likely because of climate change, which impacted the coral's ability to fend off sort of decrease its immune system. And because of this, there's a major problem with Aspergillus infections in sea fans, which are, of course, an animal just like us. So with that, I want to turn it over back to Dale to introduce Peter to us, who can talk a little bit more about Aspergillus and its effects.
Thank you, Logan. That was very good. We'll move on to our next presenter, Peter Lenders from the Center for Agriculture and the Environment in Holland. And Peter is a senior advisor at CLM Research and Advice, and it's an independent organization that stimulates sustainable agriculture by innovation, debate, research, and advice, where he is an ecotoxicologist specializing in sustainable crop protection and pesticides. At CLM, Peter has developed a tool for farmers, advisors, and policymakers that is the environmental yardstick for pesticides. Peter is also studying new and emerging problems related to pesticides. Two clear examples are the impact of pesticides on bees and the link between public health and resistance of non-target fungi. I would just like to mention that we're honored to have the head of CLM with us in Minneapolis this morning, listening to the webinar, Voter van der Wegen, and Voter has been a long-time friend of IATP, and we're really glad that you're able to be with us. But now let's turn it over to Peter, and also a reminder to, if you have questions, be sure to start submitting them. Thank you. Peter?
Yes, thank you, Dale, for your introduction and for the invitation to speak this morning, although it's afternoon in the Netherlands. I'll talk today about the azole fungicides, Aspergillus, and preliminary hotspots that we've been looking for. And actually we do this work together with some CLM colleagues, which you already mentioned, Wouter. But we also closely work together with the Radboud University Hospital, Radboud UMC, with Paul Verwij, who is really a specialist on the medical side of these problems. I'm an ecotoxicologist, but Dale, you asked me also to tell something about the human health part, and I'll come to that. First, let's have a look at the resistant fungi. They are a new risk for humans, and in the Netherlands and also worldwide there's a lot of attention on human health related to antibiotic resistance. Measures are taken, but there's very little attention so far on human health and fungicide resistance. And Paul Verwij, he showed already in 2010, he stated to the Dutch press that aggressive infection with resistant fungus could leads to death in Dutch hospitals. And Wouter van der Weyden with colleague Van der Wal, they wrote this paper in the Farmers Journal about resistant fungi being a time bomb. And we will focus today on Aspergillus fumigatus. You can see it cultured in the lab on the right hand side on this slide. So let's step to the human problem. There is an illness called invasive pulmonary aspergillosis, and that means that Aspergillus is able to enter our lungs. It enters our lungs daily, but we can cope with that unless we are very ill. So people with a bad immune system, there this fungus can grow in the lungs, especially. You can see it on the left hand side on the picture of the radiation of the lungs, and you can see the white spots is the growing mycelium. And of course doctors they treat the patients when they have this invasion, and they have a number of azoles available, like itraconazole, voriconazole, and posaconazole. They can use it, but these medicines don't always work. Let's just have a look at the kind of diseases that are coped with and the kind of persons that can be affected. Allergic people can be affected, but also people suffering from cancer, of leukemia, a number of diseases. People can have this Aspergillus disease, and it can be lead to death. And in the Netherlands, Paul Verwey has done a big study on this, and I'll go to the next slide, where you can see that mortality from Aspergillus is low in the allergic population, but it is high, about 50% for the chronic invasive aspergillosis, but also acute invasive one, people having cancer of other diseases. On the right side, you see in red that people who have got this fungi and they have the resistant one, you can see that mortality is almost 100%. So if you get a resistant Aspergillus when you are weakened by these serious illnesses, yeah, you will most probably die. Just an example of 8 patients with this azole resistance is seen in this table. You see different people with different kind of diseases. Most important is here is that you can see 4 patients actually having this, all 8 have this resistant Aspergillus, and only 1 survived. And you can also see that there has been resistance from different medicines. Okay, let's take the next step, because an analysis was made by Paul Verwey about the new resistance mutants arising, and the first one arose, as you can see in this slide, in 1998. A second one was found only once, that was in 2006, and a third one arose in 2009, and especially the last one is increasing very fast. And on the bottom you can see that in this period more and more DMI, fungicides, azoles have been used in agriculture. So there seems to be a relation. We just finished in the Netherlands an international conference just yesterday finished, and there was a lot of discussion on this, because some people say these fungicides were already introduced earlier, so they debate, there is some debate on this relation, but it's well, it's interesting to see, and it's also a reason for us to go into it much further. So I'll take you first to some more information about all this azoles shown here and their introduction years over the years, and also you can see in 1998 the first clinical tandem repeats. They call it tandem repeat 44, but that's the mutation which is really has arisen then and has given much trouble. Let's have a look at Europe, because first scientists thought this is a real Dutch problem. But as you can see here, in a number of hospitals throughout Europe, the azole-resistant fumigatus is found, TR34, but also TR46. That is the red one, which is found in 3 countries, and the other one is found in a lot more countries where measurements are made. So it's really an increasing problem. And I'll just take you shortly to China, India, and Iran, because especially TR46 is found there as well, and the genetic background of the ones found there is very comparable to the European one. So there seems to be a worldwide problem. Okay, I just want to shift to for some specific questions that we have been trying to answer, because it's of course important to know where do these azole-resistant strains originate. So just 4 questions I'll shortly go through with you. Where do we find Aspergillus? Where are these azoles used? What amounts are used? And where are optimal conditions for this resistance? Let's go to the question, where do we find Aspergillus? And believe it or not, everywhere. You find it in air, you find it in animals. It was also found in the flowers outside the hospital. So really you find them everywhere. It's a saprophyte and it can grow on compost, etc. But you also find it in houses. Next question, where are azoles used? Well, to be honest, they are used everywhere as well. Of course, we just talked about the crop protection where they are used, but they are also used, for instance, for fruit conservations, like mandarins and oranges, wood conservations, and even old film conservation as a biocide that you see left below, also for wood conservation, well, many places. So the third question we asked, what azole amounts are used? In the Netherlands, in agriculture, large amounts are used, winter wheat, but also, for instance, in the bulbs, the tulips, etc. And you see here the different fungicides, azoles that are being used. I won't go into detail, but it's clear that large amounts are used to get rid of the fungi. There are also other uses in the Netherlands, but little is known. In Dutch healthcare itself, about 400 kilograms, but other use, the amounts are not known. Okay, one remarkable observation, and Dale already told something about that too. We can see now that in the Netherlands, azoles are promoted as a growth stimulator. Here you have some Dutch words on it, but what they actually say is that azoles will help you even when there is no fungus in your maize, corn, so use it. Well, I think it's worrying because you shouldn't promote these kind of fungicides only for better growth. I don't think that's very sensible. That's a remarkable observation. But the final question I want to deal with is where are optimal conditions for resistance development, and that is not everywhere, because there has to be presence of Aspergillus, there has to be azole pressure, but there also has to be high moisture and high temperature and presence of oxygen. So at CLM, together with Rob say, we have been looking at hotspots. We call this a hotspot, where you find all these characteristics together. So let's search for hotspots, and first we look at the composting material you see here, and the above three graphs show you in fact the starting material. On the left you see grass, in the middle you see leaves, and on the right hand side you see wood chops, and that's all used to get to compost. In large company does this work. And what we found is that, for instance, in the leaves in the middle you see no Aspergillus, green, and also no resistant Aspergillus, the red one. But in the grass you find loads of Aspergillus. The green one is the asexual one, and the red one is the resistant one. And we also found some sexual, a small amount of sexual Aspergillus, which is very important, as it also you just heard that during the sexual recombination there's a chance that you get this resistance. Okay, after composting, the lowest figure you see just a very small amount left of asexual and resistant and non-resistant Aspergillus. So no azole resistant strains left here, but and the sexual strain was not reproducible. This was only a preliminary study, but we got some indications, and we also looked, and I'll show you that in the next sheet, we also looked at the bulb waste, peel waste of bulbs, because that's also put in compost. And what we found there was quite a lot of both resistant and sensitive Aspergillus. As you can see in the above picture, you can see that all the Aspergillus present was in fact resistant in this case. It's only one sampling, so the observation is just in its beginning. But in this peel waste, you see a lot of proteconazole, which we found. We also looked at an ecological farm and no fungicides were used, the bottom, and there you see hardly any Aspergillus, a small amount, and you see no resistant Aspergillus. Okay, we're going to finish on this. We got some indications. You see that Aspergillus was found in different predicted locations, both sensitive and resistant strains, sometimes only resistant strains even, and sometimes we hardly found any Aspergillus. For instance, the dry straw of wheat, we didn't find it, and also in ecological farming, we did not find it. But we had a few samples and a few replications. How to reduce the risk of fungicide resistance? We think it's necessary to identify more hotspots, to analyze mechanisms, and to try to eliminate these hotspots in different ways. And we also found out or think that it's very important to cooperate internationally and also advise registration authorities. I will tell a little bit more about this international cooperation, and then I'll give it back to Dale, because we had this meeting in a very old building, the Royal Dutch Academy of Sciences, just the last few days, with an interdisciplinary scientists from both agriculture and the medical world, which was extremely interesting. And there were some notes made by these people. It's not only the crop protection, it can also be material preservation, or even the Aspergillus therapy itself, which could stimulate this azole resistance. The exposure is of course quite important. There was some discussion on this exposure. Do you need high or low concentration? Secondly, a number of questions were of course asked: selection pressure, resistant mechanism, what's the role of sex, and also especially the fitness costs. It looks like that Aspergillus has no fitness costs of getting when it gets resistant, which is quite worrying. Finally, the people of the conference also said that looking for hotspots is very important, but also unraveling the genomics of the population of this fungus. Okay, this is what I wanted to tell you, and I'm going to switch back to Dale, and maybe hopefully can deal some questions with you and have some discussion. Thank you.
Thank you, Peter. And also I want to thank CLM for really taking an important lead in this work and bringing all this information to us. A reminder, if you have questions, you should submit them. I don't think my screen is available, but you'll see that there's a small box for questions. Just type it in and send it, and we'll be able to share your questions with everybody. I want to start with a question to Logan. In your report, you described 2 case studies about resistance, and this morning we've focused on azoles, but I'm wondering if you could talk a little bit about the other fungicide that you were looking at.
Certainly. The other case study that we looked at was an example that we selected, I think, Dale, because it was sort of your first introduction to these problems of fungicides that you mentioned earlier, a conversation you had with someone who was flying fungicide application routes from their plane. And what they were applying was a different family of fungicides from the azoles that are called strobilurins. And these strobilurins are a modern fungicide. They're very effective. They have some problems with resistance in the crops they're used most frequently in, mostly soy and wheat. But particularly in wheat, they have shown substantial effects for manifesting extra growth and extra yield. The reasons for that are still somewhat unclear, but because of those results, the manufacturers of these fungicides decided to apply them for the same purpose in other contexts. So they decided that they would also increase yield in corn, and because of that, here in the Midwest of the United States, they have been applied in very large quantities over really vast landscapes. This is obviously a problem if you're thinking about the possibility of developing resistance. It may not be a problem for public health. Strobilurins are very rarely used in medicine. I think because they're fairly bad for people in addition to fungi. But the problem with these fungicide applications in corn goes even deeper than that. Well, it seems conceivable that there's some benefit in terms of yield increases. That benefit is almost always smaller than the benefit or than the cost of the fungicides themselves and the application. So farmers who are doing this are not just increasing the likelihood that the strobilurins that they're using will become useless in the future. They're also losing money directly, and so we talked a little bit about that as a major risk for fungicide resistance going forward in the paper that came out from IATP. Does that answer your question, Dale?
Yes, I would say that in periods of high prices, the pilot I was talking to, this is my source, right? But he said that the farmers were reporting yield increases in the range of 6 bushels per acre. And in periods of high commodity prices, it paid off. But as we know, those moments come very rarely. So, but yes, thank you, Logan. I have a question here, which is, if this explosion of fungi also appears in wild populations, we must fear that the agri lobby argues that these are natural processes. How can we know and show who have caused these explosions? Peter, anything come out regarding that at your conference, at your symposium?
Yes, certainly. Of course, there are the wild types are present in nature, and of course there are all sort of kinds of precious stress on these fungi which can create mutations. But it is clearly shown also in the lab that the azoles, whether they come from pesticide use or wood conservation or whatever, they have caused these mutants which are at risk. So there's no argument about this. Also, everybody agrees that the azoles have induced these two mutations which I mentioned. So there's no discussion.
All right. Were there other recommendations that came out from the symposium of steps that we could be taking to help reduce fungicide resistance, or just to understand it better?
Yeah, I think both things were mentioned. Of course, the scientists they really want to understand the complete life cycle of this Aspergillus better, to find out what can be the reason and what can be the source. Because if you know the source, you can try to eliminate the source and also eliminate the spreading, because the spreading is really a big issue. Aspergillus goes easily through the air and can get everywhere through the air, also in everybody's lungs. So it's important to look into the mechanism and the life cycle. So that was an important issue. But another thing was, and I think most of the people present agree on that, if we need fungicides like azoles to control fungi on all sort of spots and also in agriculture, you have to be very careful by using it. So you have to make your choices, and you also have to think about alternatives. Of course, we know that you have resistant varieties of all our plants. So you also will have to invest in developing these resistant varieties. So then you don't have to use a lot of chemical fungicides.
Very good, thank you. Our next question is, to what extent are farmers using fungicides to increase corn yields, aware of the risks inherent? I don't know, Logan or Peter, who would like to take that? I can tell you from my own experience that farmers are given almost the exact opposite information about the risks and more about the potential benefits from the use of fungicides. But which of you would like to have anything that you'd like to add to that?
Yeah, well, it's a bit different here, but I think in the Netherlands, the companies, the pesticide companies, are aware that there is a risk. So what they do now is they sell a lot of combination fungicides. So they put 3 different fungicides with different mode of actions in one product. And that's the way that they say that they can prevent resistance.
If I can add to that just briefly, here in the Midwest, where farmers are using corn for yield increases that are often illusionary, the part of what's driving that is the sort of information on that's provided with the fungicide, and a lot of where that is coming from is directly from the fungicide manufacturers. And so even though in an academic setting it's pretty clear to researchers who work with farmers that there are some problems with this. Much of that information is not getting to the farmers themselves, who see the handouts that come with the fungicides as fairly authoritative. They're sort of signed off on by the EPA, although the EPA does not actually have any direct input into what information goes into those handouts.
Thank you, Logan. We have a question for Peter. One of the slides you showed from the meeting in the Netherlands showed migration from hotspots. Can you say more about migration?
Yes. Logan already told about the large number of spores that are always, of course, coming from these fungus, and Aspergillus is now is a good example of that. When there are spores formed in a hotspot, they will spread throughout the air over long distances. So can really these spores can travel quite a distance, and can come down again further away. So it's as soon as there are spots where you have Aspergillus, where you have resistant Aspergillus, these resistant Aspergillus can also easily travel away from to a hotspot into the environment quite a distance away.
Okay, thank you. Would anybody like to comment on how fungicide resistance might be affected by climate change?
I can touch on that briefly. So one of the problems that we see with climate change is right, changes in growing year length, right? How long can these fungicides stay active in the environment? And of course, the longer that is, the more generations they can get in a single year, the more likely that you are to sort of see the initial mutant that's resistant to a fungicide. Additionally, climate change impacts the ability of fungicides to move long distances, right? They can, instead of just initially being in one place, right, they can increase the range that they are able to survive. That movement across long distances can often facilitate, along with you know, sexual parts of the fungus life cycle. Those can both sort of increase the possibility of developing resistance. Does that answer the question?
Yes, that was helpful. I want to go back to the issue of what the questionnaire is calling the fungicide cocktails, the mixing of two or three different fungicides together to deal with resistance. And the question is, are these fungicide cocktails effective in guarding against resistance?
I have a very interesting remark about that, because we discussed it in Amsterdam at the conference as well. And agronomists, they say, well, we do always cocktails now because they are effective. And they also told the medical people, you have to do cocktails, because why don't you do cocktails? So in the medical world, a cocktail is less normal than in the agronomy world. That was striking. Well, so as far as we know from the agriculture world, they say that cocktails is better because then you have this different mode of actions at once. So that keeps the resistance down. But it's quite new. So I've asked the question at the conference as well: what will bring this in the long run, or will there be a sort of combination resistance? But the specialists said, well, we think it's the best way to go.
Thank you, Logan. I'd like, yeah, jump. Yes, go ahead.
Yeah, that was a great response. I just want to, I got a note here from Wouter that is an important thing to touch on, especially the question that we got about sort of the distinction between natural processes, right, and who is responsible for these new resistant and other emerging fungal diseases. And it is important to distinguish between the natural extinctions and diseases that we're seeing, right, in bats and frogs and other amphibians, right, which has causes ranging from climate change and trade, but often those fungi are not fungicide resistant, whereas these resistant fungi that are causing so many problems in human health and in agriculture and livestock, right, those are driven also by trade and also perhaps by climate change, but really by fungicide use, right? Each application of fungicide is going to be a selection event that's going to make these problems worse, whereas we very rarely try and use fungicide even to help the extinctions in bats and amphibians and in other wild species. So that's an important distinction to make that I hope is clear to all of the people listening out there.
All right, thank you. In response to the cocktail answer, so this is still theoretical and not evidence-based.
Yeah, well, it depends on the time scale. I mean, the last few years in Holland, it are all cocktails, and of course tests have been done by the industry showing that a cocktail works at this moment works better than a single application, and even better than single fungicides, and then well, changing over to the next fungicide and changing over to the next fungicide again. So there is evidence-based, but of course those tests are always for a certain short period of time. So I meant that if you look over 10 or 20 years, will then still this cocktail strategy be effective? That's I don't know. Could be a new problem arising later on.
If I can add on to that just briefly, there are both a lot of theoretical studies on the difference between single fungicide versus cocktails, but also there are a wide variety of empirical studies that Peter is exactly right. They're generally short time scales. It's hard to know how long those effects will persist, and whether we can keep developing new fungicides to sort of keep up with the multi-resistant fungi that come from regular use of mixed fungicide cocktails. But there's also an impact those studies can also show the costs of, if you're mixing an azole with a much older fungicide, you're getting both the minimal impacts on the environment and human health of the newer, more complex fungicide, but also the older one that you're mixing to help increase the impact often has much more serious impacts for the environment and for humans and for wild species as well.
Thank you, Logan. And with that, I would like to wrap up our webinar. And I want to thank Logan and Peter and CLM for this great presentation, and thank all of you for joining us. You'll be hearing from us shortly with the follow-up information. So thank you. Goodbye.